Imagine the computer you're watching this on was run by cells. Well, this is essentially what a biocomputer is. A biocomputer is a computer that uses biological substances to carry out computational tasks. If you want to learn more about what a biocomputer is, I suggest watching my Introduction to Biocomputing, which I linked below. So, if you've ever worked with a computer, you know there's a lot more to it than just pressing the on button and the off button. And the same goes for a biocomputer, except for there's even more steps and processes, since we are transforming biological substances to do stuff that usually human-made substances do. So that's why I decided to simulate a genetic NOR logic gate, because I wanted to learn about how the different processes work in a biocomputer compared to a regular computer. So first you need to understand what a logic gate is. A logic gate is something in Boolean logic, which is a form of algebra, that tells you to perform a certain function. So for example, if you had a light switch and you said that if light switch A was turned on and light switch B was also turned on, then the light bulb would turn on. This would be an AND or an AND logic gate. Now, a NOR logic gate is created. It's a combination of a NOT and an OR logic gate. The cool thing about a NOR logic gate is it's a universal logic gate, which means it can be combined to form any other type of logic gate. And how it works is it would be like if both of the switches had to be down for the light bulb to be turned on. That's essentially how this works. So first, as you can see, you need input A and input B. In this case, input A and input B would be proteins, or what we call transcription factors. The input A and input B are then assigned to a promoter to create what we then would need as protein X and inducer 1. So protein X will be created in the ribosome binding site right here, and inducer 1 will be created in the repressor binding site. Now inducer 1 is going to be producing proteins that we want, but we're actually going to repress those because we don't want them in order to perform our function. So then after that, the protein X is going to tell inducer 2, which is also another output of input A and input B, to not make proteins either. See, since it's a NOT OR, then we want both of them off. Inducer 2 is going to be over here and created at the ribosome binding site number 2. Now we have created the OR function and the NOT function of our logic gate. So all we need now is the switch. And the switch is going to be created from what we call a genetic repressing, or a gene repressing regulation. And it's going to be caused by protein X to repress inducer number 2. So as you can see, now we have built our logic gate. So how this works is we're going to click run. And here you see we have all of our values. So we're going to go to initial values. So in order for this to work, we need to have something in transcriptor 1 and 2, because those are our inputs, right? Oops, not that one. So I'm going to just put it like up to here. So now I'm going to click Save Values, and I am going to look at our graph. So the first step is, since our graph's like super cluttered right now, is to go to the legend, click None, and then only select code 1, which is the block that contains protein X. And then we're also going to want to click RP1, which is the repressor. We're going to click Update Plot. Now, as you can see here, we have the values. This is our repressor, and it's doing its job. It's repressing the protein production. And then we have code 1, which is the protein X, and as you can see, it is being produced. Now, if we go back over to our values, you can see that if we change the inputs, the graph will change. if you watch the protein X. And so that's essentially how a NOR logic gate works.